Hello everyone, this is Muhammad Arbas from Shivankeshwara College of Engineering. In this session, I will be discussing about cardamom plant disease detection approach using EfficientNetV2. I will be discussing introduction part, literature survey, proposed system, cardamom diseases, data set description, proposed methods, equations, structure of MB convolutional and fused MB convolutional, experimental analysis of the system, final results, conclusions, and what are all the references I have preferred to prepare these slides. Let us come to the introduction part. Cardamom is a queen of species. It is indigenously grown in the evergreen forest of Karnataka, Kerala, Tamil Nadu, and northeastern parts of the India. India is the third largest producer of cardamom. Cardamom is a plant species that belongs to the ginger family and is native to the Indian subcontinent and Southeast Asia. It is also grown in Sri Lanka and other parts of the world. There are mainly two types of cardamom, green cardamom and black cardamom. Green cardamom is also known as true cardamom. It is grown in Sri Lanka and India. Black cardamom is also known as large cardamom, which is used as a flavoring agent and it is grown in eastern parts of Himalayas. Cardamom is also used in medicines including allopathy and Ayurveda. Cardamom is known as aromatic flavor and is used in variety dishes including curries, stews, and other dishes. It is also used in coffee, tea, and beverages. Various disease and pests affect the growth of cardamom plants at different stages and crop yields. There are several diseases that can affect cardamom plants including fungal disease, viral disease, and bacterial disease. Here are few examples: leaf spot, root spot, fusarium wilt, mosaic virus, bacterial wilt. To prevent and control disease, it is important to practice good plant hygienic such as removing diseased plant materials and avoiding overcrowding of plants. To address the challenges while detecting plant diseases, image processing using an automatic plant leaf detection approach is essential. In recent study, computer vision and machine learning based techniques were developed for plant leaf detection. Let us come to the literature survey part. A author by name Rashmi N has published a, a machine learning technique for identification of plant diseases in leaves, which was published in the year 2021 by IEEE. The methodology used is artificial neural network, considered classification as one of the most dynamic research and application areas. The neural networks was trained by propagation algorithm. Neural network represent a mathematical model of information processing and can be trained and learned the same as human brain does. An author by name Pushpakara Sharma has published classification of plant leaf disease using machine learning and image processing techniques, which was published in 2020 by IEEE. In this methodology used is plant leaf detection and classification based on CNN, which is nothing but conceptual neural network with LVQ algorithm, plant disease detection using image segmentation and SVM techniques. The author by name Swatika R has published disease identification in paddy leaves using conceptual neural network based on deep learning, which was published in 2021 in third international conference on intelligent communication technology and virtual mobile network. The main methodology used is an enhanced version of artificial neural network is the conventional neural network which is greatly used in image classification and speech recognition. Feature extraction by SVM model and k-means clustering algorithm is applied to cluster the pests according to their types, which is resulted in three major clusters of pests. Let us come to the proposed system. This proposed system is concentrated on two diseases: cardamom plants, Collectridium blight, and Philosticta leaf spot of cardamom, and three diseases of grape, blackroot, esca, and Iseriopsis leaf spot. U2net is used to remove unwanted background of an input image by selecting multi-scale features. Here we will be using EfficientNetV2 because EfficientNetV2 is a family of convolutional neural network models developed by Google in 2020. These models are designed to be more efficient and accurate than previous state-of-art models such as original efficient net models. Cardamom plant disease detection approach using efficient NetV2 model. Efficient NetV2 model is several advanced techniques such as scaling attention mechanism. The idea behind scaling is to balance the depth, width, and resolution of the network to achieve better performance. Attention mechanism helps the model to focus on the most important features. A comprehensive state of experiments was carried out to ascertain the performance of the proposed approach and compare it with other models such as efficient net and convolutional neural networks. These are all the cardamom diseases. As shown here in first picture, cardamom collectridium blight, which is on leaves, it is yellowish color spotted spots are present. Another in figure B, cardamom Phyllisticta leaf spot, and another in final figure that is shown at last, which is healthy cardamom. Now I will be discussing about dataset description, image capturing setup. Real-time cardamom plant leaf images were captured in farmland using different mobile phones or cameras or different capturing devices. Let us come to here the image is shown, image capturing setup. We have to capture the images which are present in the garden. Multiple images we have to capture. This is the real time image capturing setup. Cardamom dataset. Image is captured under farm field scenario without technical means. It reduces the size of original image captured. Plant village dataset. Plant village is freely available dataset that is widely used in field of plant disease detection. It contains over 54,284 images, all of which were annotated. Available grape datasets was also used. Proposed methods. The proposed system has two methods, background removal and classification models. Now let us come to the background removal. Cardamom plant leaf image are of RGB, red, green, blue, which are the basic colors. The background removal approach is widely used in the study of U2net, and this equation is used for the removal. This equation consists of residual connection and surplus connection. Residual connection combines the surplus connection, which combines local attributes and multiple attributes as shown in the equation. The second part of the NetV2 architecture is a 5-stage decoder that uses deleted version of residual surplus connection. It shows the pixel unique quality. When pixel is the smallest unit of any image. When pixel values are more, then image quality will be good. Here we will be using the equation u of fm of x plus fm of x. If an identity mapping were optimal, it would be an easier to push the residual to 0 than to fit the identity mapping by a stack of a non-linear layers. First, weight layer will be directly sent to application function ReLU. Then ReLU will be XORed with then weighted layer will be XORed with ReLU. This is an residual U block diagram. Let us come to the schematic representation of background removal by using NetV2. First, we will be capturing the image of plant. Then we will be downsample the feature maps. Then this feature maps will be encoded into high resolution features. Then we will be removing the color. At last, we will be comparing bitwise. We will be doing bitwise operation. Let us come to the classification model, which was another proposed method. In this, we will be using conceptual neural network. Conceptual neural network consists of various layers such as convolutional, pooling, and fully connected layers. Conceptual layers extract the dataset of future maps and known as color and edges of the input images. The pooling layer reduces the possibility of overfitting by minimizing the spatial dimension and convolution. The one then one or more fully connected layers defined, which is attended the network by connecting all the previous layers neurons. Finally, fully connected layers predicts the class labels, where softmax activation function is used in the pre-trained models which are used in this work. These are all the equation. For residual and surplus connection, we will be using u of fm of x and fm of x. Here fm of x will be, that is nothing but weighted image will be XORed with application function along with the weighted image. And here it is shown that loss is given with fusion and slide. This is the structure of MV convolutional and fused MV convolutional. First diagram represents the MV convolutional. In MV convolutional will be given to depth convolutional and it is squeezed and excitation, and at last it will be XORed with what are the input will be giving. In the fused MV convolutional, we will not be using depth-wise convolution. The apart of that, all the features will be same in this MV convolutional, fused MV convolutional. As compared to MB convolutional, convolutional MB convolutional will be better because it is having less layers and it is easy to process all the given inputs. Let us come to the efficient net. Efficient net is a family of CNN proposed. Efficient net is used to scale up the CNN. It uses mobile inverted bottleneck convolution, that is MB convolution, as baseline network and scale up this network as efficient net. This schematic representation represents proposed cardamom plant leaf disease detection approach. First, we will be capturing the image which are present in garden. We will be taking all the images of the cardamom plants. Then we will be removing background by using efficient NetV2, U2 NetV2. After removing the background, we will be giving to image resizer. Then train deep learning model is applied. Then classification of the leaves are done. In classification, we will be uploading the healthy image of cardamom plant. In classification, the healthy and the disease the affected cardamom plant leaves are compared. If it is caused by any disease, then it will be showing that which disease it is caused, or else it will be showing that it is not caused by any diseases. This proposed system consists of two phases, training phase and testing phase. In testing phase, it will be doing all, it will be comparing that real image of the healthy image of the cardamom plants. This is the experimental analysis of the system. Here it is shown how it will be comparing in this, how all the features are compared. This is the confusion matrix of the cardamom plant datasets. This is obtained by using external testing. First diagram is used by EfficientNetV2 for cardamom plant dataset, and in this it is shown healthy phyllostickta leaf spot that is caused by disease, and coletrichtum blight, which is also caused by disease. And this is also a confusion matrix, but it is for grape plant dataset. Last it is shown for cardamom plant dataset, but it is this slide is discussed about last grape plant dataset. Let us come to the final result. For cardamom plant, CNN value is 87.59, efficient net is 87.02, and efficient net V2 is 91.42. This is nothing but if we use the CNN, that is convolution neural network, we will be getting 87.59. But if we use efficient net, we will be getting 87.02. But if we are using efficient net V2, we will be getting 91.42. That is nothing but detection accuracy. For grape, we will be getting 94.60 in while using CNN. If we use efficient net V2, we will be getting 94.62. This is the explanation what is given, what I have explained. The efficient net V2 L model outperforms the other two models on cardamom plant dataset with 91.42. When compared to efficient net and convolution neural network, efficient net V2 is better because it will be giving accurate detection. A wide set of experiment was conducted on neural network, convolutional neural network, net and efficient net V2 models. Employing U2 net and with efficient net V2 outperforms for real-time cardamom plant leaf disease detection with 98.26 detection accuracy. When we use U2 with efficient net V2, we will be getting 98.26 accuracy of detecting plants disease of plants. The conclusion is an efficient plant leaf disease detection approach is essential to detect plant diseases in real time. Segmenting and detecting diseases in real time image is challenging task as the images are associated with other factors such as background of image, environmental factors such as lighting and angle of the capturing conditions. The dataset can be enhanced by collecting images of cardamom diseases with nutrition deficiency in future work. Furthermore, the model can be extended to identify the severity of diseases and nutrition deficiency. And this is the reference I have preferred. Image recognition technology based on deep learning, which was published by X Wang and Z Liu. Leave disease segmentation and classification of Jatropha carcass and Pongamia pinnata biofuel plants using computer vision based approaches measurement volume, which was published by Chauhan and UP Singh and U Sharma and S Jain. The next reference I have prepared is identification of cash crop disease users using automatic image segmentation algorithm and deep learning with expanded dataset, which was published in 2020, which was developed by M. View and L. Wang. And the bacterial fragment optimization based radial basis function neural network and classification of plant diseases was published by UP Singh and US Jain. UP Singh and US Jain have published two references which I have preferred. At last, multi-layer convolutional network neural network for the classification of mango leaf infected by anthracnose disease was published by S.S. Chauhan and S. Jain and UP Singh, which was published in IEEE conference. Thank you.